The Thammasat University Drug Discovery and Development Center was established in 2013 with the support of the Thai Chem Kang project. The DDDC aspires to become an internationally recognized center for drug research that transforms multidisciplinary basic research into innovative translational products for major infectious and non-infectious diseases. Our mission is, first, to discover and develop new chemical entities derived from natural products through interdisciplinary research to fight against these diseases, and second, to offer our resources, facilities, and services to support other like-minded researchers in their drug discovery and development work. Our research center provides various services, including access to a wide array of lab instruments, lab test-based service, and research-based service spanning 5 different research fields: medicinal chemistry, pharmacology and molecular biology, animal imaging and toxicology, microbiology, and finally, clinical research. At our department, the medicinal chemistry unit focuses on lead discovery of compounds derived from natural products for targeting specific molecular entities or diseases. The phytochemistry subunit investigates herbs used in ethnomedicine, identifying the key bioactive components within them, and may also go on to chemically modify these components as a part of further research and development. The process of phytochemical research starts with the selection of plant material to be studied using ethnomedical knowledge. The plant material is then collected and prepared for further research. To account for variability between plant specimens, analysis of certain marker compounds is performed as a quality control of the plant extracts. Taxonomical identification of plants is done based on morphology, and the chosen voucher specimens are added to the herbarium. The plant material is then dried in a hot air oven and then processed by a mill. Extraction is carried out using one of several techniques, including decoction, maceration, ultrasonication, soxhlet extraction, and microwave extraction. The phytochemical constituents of the plant are extracted and isolated so that their chemical structure can be elucidated. The extracting solvent is removed using a rotary evaporator or freeze dryer, leaving behind only the phytochemical constituents of the plant. Next, phytochemical screening is performed using TLC. Isolation of active components is carried out by column chromatography, preparative HPLC, or preparative TLC. Then, the molecular structures of these active components are characterized through spectroscopic techniques, such as NMR, IR, and MS. After all of this, it may be possible to improve the biological activity, pharmacological activity, or physiochemical properties of some phytochemical compounds via chemical modification. This process can include changing the structure, configuration, form, biosynthetic ability, or moiety groups of a compound. The Pharmacology and Molecular Biology unit of the DDDC carries out preclinical studies, both in vitro and in vivo, using pharmacology and molecular biology techniques. The cell cultivation facility, along with several analytical instruments, such as our microplate reader, real-time PCR machine, in-cell analyzer, flow cytometer, and FPLC, have been set up for our own research as well as for the purpose of providing our instrument and testing services to others. Now further along the drug discovery and development pipeline, the work of the Pharmacology and Molecular Biology unit begins with receiving crude extracts or synthetic compounds from the medicinal chemistry unit or an outside collaborator. Working in the cell cultivation system, PPE is worn at all times and sterile technique is strictly followed in order to maintain system sterility, protect lab personnel from infection, and to prevent the spread of biological agents. At this facility, cells are cultured in a CO2 incubator. The CO2 incubator is used to create an environment optimal for cell growth. When a cell culture is ready, an inverted microscope is used to observe various characteristics of the culture, such as cell number or cell morphology. Continuous cell cultivation is performed in the biosafety cabinet. Once the cells are ready for testing, they are transferred into a 96-well plate and then introduced to the compound being studied. Then, the interaction between test compound and cell culture is observed by performing an MTT assay or using Presto Blue reagent. Afterwards, the data are analyzed by computer software. Investigation of test compounds at the cellular or molecular level is done in the Pharmacology and Molecular Biology lab. The in-cell analyzer and flow cytometer can be useful to study the effects of a compound at the cellular level, such as alteration of the cell membrane or cell proliferation. For observations at the molecular level, real-time PCR is used to study gene-specific expression, while the protein study system of instruments is used to study protein-specific expression. After this, the specific protein can be selectively purified by FPLC and be used in further experiments. Animal testing is used in many steps of preclinical research in the process of drug discovery and development before clinical studies on humans begin. With a state-of-the-art laboratory for animal research, the DDDC provides the in vivo imaging lab for hosting research projects by academic staff as well as outside collaborators and entrepreneurs. The Animal Imaging and Toxicology Unit focuses on the application of molecular imaging techniques to investigate the distribution of test compounds and their targets of action in small animals. Such imaging is accomplished using the facility's PET SPECT CT scanner and the optical in vivo imaging system. This unit is also equipped to carry out in vivo toxicity studies, allowing us to assess the safety of test compounds. Our lab facility is managed following the AAA-LAC International Animal Care and Use Standard, as well as the National Radiation Safety Standard. This lab space is divided into 4 zones: the Anti-Zone, Animal Holding Zone, Radioisotope Preparation Zone, and the Animal Imaging Zone. During the process of in vivo molecular imaging, animals are taken from the Thammasat Laboratory Animal Center to the Animal Holding Zone, and then conditioned for testing before research begins. Here, researchers wear PPE at all times to ensure safety and a well-maintained lab environment. In the case of performing radioisotope imaging, a radiation survey meter as well as an OSL dosimeter are used. The radioisotope solution is prepared according to animal body weight and measured using a radioisotope dose calibrator. Then, the radioisotope solution is injected into the animal. Before imaging, the animal is anesthetized and then placed inside one of the imaging systems. In the case of using the optical in vivo imaging system, 4 imaging modes are available: fluorescence, luminescence, X-ray, and radioisotope, using Carestream in vivo FX software. The anesthetized animal is placed inside an imaging cabinet. The imaging system is suitable for preliminary detection and monitoring of drug therapy, drug distribution, and drug targeting in live animal, producing 2D images as data. When highly advanced molecular imaging techniques are required, we have available a PET SPECT CT scanner that produces high-quality images of deep organs and tissues of the animals. The Alibra imaging system we employ has 3 subsystems, known as PET, SPECT, and CT. PET and SPECT imaging provides functional information of cells and organs by utilizing radiopharmaceuticals, whereas CT imaging provides precise anatomical information like bone structure. When using the scanner, the anesthetized animal is placed on a stretcher at the animal preparation unit and is administered isoflurane via nose cone for the entirety of the imaging process. Our PET SPECT CT scanner provides 3D images as well as qualitative and quantitative data analysis using PMOD software. For in vivo toxicology work, the Animal Imaging and Toxicology Unit works in collaboration with the Laboratory Animal Center of Thammasat University, as well as the Center of Excellence in Pharmacology and Molecular Biology of Malaria and Cholangiocarcinoma of Thammasat University. After the safety and therapeutic potential of a health product has been sufficiently demonstrated by preclinical research, further research can be initiated in human participants. These studies, known as clinical trials, are carried out in 4 phases, simply referred to as phases 1, 2, 3, and 4. Phase 1 is usually the first stage of testing to be done with healthy humans. This phase is meant to determine the safety and maximum tolerable dose of an investigational product before any further human testing may be carried out. The Clinical Research Unit of the DDDC provides fully equipped facilities staffed with medical personnel used for conducting Phase 1 clinical trials of low-risk health products. For example, research may be conducted on the pharmacokinetics, pharmacodynamics, and bioequivalence of locally made health products. We also provide clinical research design and ethical consultation services. At the beginning of a clinical trial, volunteers must sign an informed consent form, acknowledging that they fully understand the study protocol, as well as any possible risks or benefits associated with their participation in the study. Clinical trials are performed with volunteers who have passed the required clinical and lab examinations, such as a blood chemistry test or physical. These checks are performed in order to confirm appropriate health conditions before any experiments are done with the volunteers. Following the preliminary examinations, volunteers are each randomly assigned to a study group. Depending on the study design, groups may be assigned to receive test compound, placebo, or a product already on the market. A pharmacist will prepare and then distribute the assigned products to each volunteer according to their respective study group. During the testing period, vital signs are checked and other required examinations are regularly performed, with the results being recorded by a medical professional in the clinical examination room. Peripheral blood samples are collected from volunteers at predetermined times by a medical technician or nurse. The samples are then aliquoted and stored according to proper methodology to ensure that volunteer privacy is protected and that the sample quality is well maintained. The samples are eventually analyzed by methods such as LC-MS, flow cytometry, or PCR. At our lab, data are collected, processed, and analyzed following clinical data management. Clinical data management is a process encompassing the whole of a clinical trial, from data collection to analysis, all done in compliance with regulatory standards. Clinical data management is very important, as it is the process by which we are able to consistently produce data that is statistically sound, reliable, and of high quality.